Hello, good morning, class 9 student. How are you? I hope you all are doing well. And the previous video was a Q&A session, and I hope that you got your answers. And yes, this is the third video of chapter 8. So let us back to the same chapter, that the third video of this chapter. And the name of the chapter, as you know, it is seed structure of seeds and seed germination. So in the previous video, we have discussed about the kinds of germination, and I think now you may get to know that there are two kinds of germination, which are apigeal and hypogeal. But there is one more kind of germination, and which is very peculiar. And let us see what these are. These are the first. Let us discuss the learning objectives of this chapter of this video lesson. Viviparous germination, condition necessary for seed germination, experiments for the necessary conditions. Okay, so these are the learning outcomes of this video lesson, and let us see that what the peculiarity of this type of germination, the viviparous germination. So this is a kind of germination in which a seed, while it is still attached to the parent plant and nourished by it, means what? That this is a different type of seed, a different type of plants. Especially, these are the plants which grow in marshy lands. So marshy places, these are very that the soil is fine, and actually that the seeds, if for example that the seed drop on that soil, so because that the soil is so weighty and fine, so maybe that there is a lacking of air. It means that they do not that the seeds do not get proper air for breathing, for respiration. So for that purpose we can say that the seeds cannot germinate. So nature has given a wonderful adaptation in such type of plants, the marshy land plants, and especially that the sea coast plants or the marshy land plants. What is that? That it is a viviparous germination. In such type of germination, that the plants have their fruits as like normal. Okay, but the peculiarity, the difference is that such fruits have seeds which grow in them. Okay, and this seeds starts germinating itself when it is still attached with the fruit. Okay, so that's why this is called viviparous germination. It means that the seeds start germinating while it is still attached with the parent plant. It's not gonna fall down. First of all, it germinates there. Okay, so that's how the first structure that comes out of the germinating seed is radical. Okay, so that the radical comes out and it grows longer. Okay, when it grows longer, it needs its nourishment. Okay, so it gets its nourishment from the cotyledon and from the fruit itself. So it gets swells up, that the radical gets swells up, and when it becomes heavy, then it falls down on the ground. Now the radical, along with its hypocotyl, along with the seed, it falls down on the ground, and then as the radical uh, develops more and form roots, so now roots are able to hold the soil. Okay, and after that the plumule grows. That plumule grows into that the tiny leaves starts growing onto it. So let us see how. Here you can see the marshland plants or mangrove plant. A mangrove plant are of different types. I can give you example: rhizophora, soneratia, heritira. Okay, so these are the plants, trees, in which you can see that this one. Okay, so this is a fruit along with its radicle. The zoom structure is given here. So you can see that the fruit is there, inside that there is a seed also, but along with it there is radicle. Okay, so the radicle is also there. This radicle grow more and more. This is the actually that I can say that the stalk of a plant. Then here that the radicle grow more and more. Okay, it swells up, and then it detaches itself from the parent plant, fall down on the ground, and then this radicle develops into roots, as you can see here below. Okay, and after that, the from the plumule, that the tiny leaf starts growing. So this is a different type of germination, exclusive type of germination, and that is called viviparous germination. It means a germination of the seed while it is still attached to the parent plant and get nourished by it. So I think you understood. This is the real picture of VV Paris germination. Here you can see that this is the fruit. Okay, so that the fruit is there, along with it it's a long radicle. Okay, and this part will will further develop into roots, and this part is, we can say that the hypocotyle is there. Okay, and then when it detaches itself from the parent plant, then the plumule will grow from this part. Okay, so this is about viviparous germination. Now the next topic, conditions needed or necessary for germination. So about conditions, we have already discussed that there are different conditions which are required for the seeds to germinate. Okay, and these are necessary conditions. What these are? Oxygen, water, and temperature. So do you know about germination? What is it? The process by which seed emerges from a period of dormancy and starts to sprout. Okay, and then converts into or develop into seedling. So this is about germination, and for that it needs these three factors: oxygen, water, and temperature. So oxygen is what? That oxygen from the air, it gets the seed needs oxygen, and without it the seeds cannot grow or cannot germinate. Why it is so? Because for all the metabolic activities, that the metabolic activities for the seeds to germinate, this will occur only when the stored food get oxygen, okay, and release energy. So for respiration, or we can say that for oxidation of food, this oxygen is needed. Okay, this is stored in the form of ATP in mitochondria. Then water. Water, this is also really important. As you know that water needs for the to lose the seed coat. Okay, and also that different metabolic activities, that because water is a medium in which all the metabolic activities takes place. Okay, so for that water is needed. And also we can say that water at in a dry seeds, actually there are 10 to 12% of water, but with this low content of water, there is no reactions takes place. Water is essential as it softens the seed coat and facilitates the necessary chemical changes in the stored food material. That's how we can say that water is needed. Water is one of the necessary conditions. The next one is temperature. So a suitable temperature is necessary for germination of seeds. Most seeds can germinate in a wide range of temperature between that 5 degree to 40 degree. But the optimum temperature or the best suited temperature, we can say that it is 25 to 30 degrees C, and that is also called optimum temperature. Okay, and in which the seeds are able to germinate because they can perform their metabolic processes. Okay, within the limits, the higher the temperature, the more it's the germination. Within the limit, it means between 5 to 40. So higher the rate of higher temperature, then higher the rate of germination. Okay, but if it is more than 40, then also that germination will not take place. And if it is lesser than 50, sorry, lesser than 5, then also germination will get affected. So these are the conditions: water, oxygen, and temperature. Now experiments. This actually experiments, I think that these are interesting topic. So let us find the first one, that how that water is necessary for germination. So for that, what you have to do, you have to take two beaker and name them as A and B. Okay, and in beaker A you have to take a dry cotton cloth, no, cotton wool or dry cotton cloth, and in the second one you have to take weighted cotton cloth. Along with that, you have to keep seeds. Okay, it can be gram seeds, pea or bean seeds. Okay, and in the same you have to put the same quantity of or equal quantity of seeds as in A, and put seeds. okay, and keep the setup for 2 to 3 days. What you will observe? What will be your observation that time? That in which beaker the seeds germinate? It is very clear that in beaker B. Why it is so? Because in beaker B there is wet cotton wool. Okay, and wet cotton wool has some amount of water in it. Okay, and seeds at the time of germination they receive such water, absorb that water, and then the seeds get sprouted. Okay, so this is how we can say that water is needed for germination, and which is not seen in beaker A because there is no water in beaker A. Got it? Then next experiment. This experiment to show that the suitable temperature is necessary for germination. So what we have to do, we have to take the same thing, that we have to take two beakers, beaker A and beaker B, and this like we have to take wet cotton wool in both the beaker. Okay, this time we are taking water also, that it means that the wet cotton cloth we are taking along with the same quantity of seeds in both the beaker. Okay, and now what we have to do, we have to put beaker A at room temperature. Okay, and beaker B in room temperature, and beaker A in ice cubes, or we can you can keep it in refrigerator also. Okay, and keep it for 2 to 3 days. And after that, what will you observe when you open, when you uh, see that what the changes occurred, or is the are the seeds germinate or not? So you will see that in beaker A there is no change in the seeds. Okay, they are as same as you have kept it. Okay, but in beaker B the seed starts germinating. So why it is so? The water is there, then why the seeds not germinated in beaker A? Now it's because the temperature is there, because there is less temperature, there is a refrigerator, it means the temperature is not between 25 to 30, and that's how the seeds do not germinate. So we can say that temperature is a necessary factor for seeds to have germinate, to get germinated. Okay, now the next experiment to show that that oxygen is necessary for germination. Yes, that oxygen is necessary for germination. Now this time what we have to do, we have to take two flasks. These are actually flasks. Okay, so we have to take two flasks, and this wet cotton wool, we have to take wet cotton wool, and name this flask as A and B. Okay, and keep the equal quantity of seeds in both the flasks. Okay, but one thing which we are going to do is that in we have to take two test tubes, and in one of the test tube we have to keep plain water, and in another test tube we have to take alkaline pyrogallic acid. Okay, so here we put plain water, and in the B1 we have to put alkaline pyrogallic acid. We have to hang it slightly carefully by using a thread, and then we have to close it by a rubber cord. After that we have to keep the setup as it is, do not disturb it, and uh, then watch after 2 to 3 days. We will see that in the flask A the seed starts germinating, they change into sprout, but here, that in beaker B there is no change. Why it is so? As alkaline pyrogallic acid is there, and which we have not poured onto the seed, but still its presence make the seeds not to germinate, because alkaline pyrogallic acid has a quality, or we can say that it is a characteristic, that it will absorb the oxygen. Okay, so the oxygen is absorbed by the pyrogallic acid, and that's how the seeds will not get germinated. But here that oxygen is already there in the flask itself, and that's how the seeds will get enough oxygen, they get proper temperature, because we have kept the setup at room temperature. And also that wet cotton cloth is also there, it means that the water is also there, oxygen is also there, and temperature is also suitable, and that's how that here in A the seeds germinated, but in B the seeds will not germinate. Okay, So now I think you understood that these factors, temperature, water, and oxygen, these are the essential factors or essential conditions for seeds to get germinated. So last experiment is three bean seed experiment. This is an interesting experiment. What we have to do that we have to take a glass beaker, half fill it with water, and that take a glass slide and take three equal sized bean seeds and tie them as shown in the figure, and keep this thing in mind that this one, that the middle one, should halfly immersed in water. Okay, then keep the setup as it is. Now what would be your observation, that which one will germinate? That the seed, the first one, that is which is on the upper region, will not get germinated because it is there in the air. It means it is getting oxygen, it is that the temperature is also favorable, but it will not get water, and that's how it is not going to germinate. Then this lower one or the bottom one, here that it does not it is getting water, that the temperature is also favorable, but the oxygen which is there in water is not sufficient to make it germinated, so that's how it will not get germinated. But the third one, that this the middle one, I can say, this middle one, it is getting that proper oxygen also, it is getting water also, and the temperature is also favorable, and that's how the middle one, the middle seed, gets germinate. Okay, so this is called three bean experiment, and which shows that water, air, and temperature all are essential for seed germination. Now, this picture is for you students. All conditions are there in A, in B, leaking water, in C, leaking oxygen, and in D, leaking suitable temperature. So, what will happen if one of these condition is lacking? Will the seed germinate or not? If you have answer for it, then I'm sure that you understood the whole video lesson. And if it is not, then do watch it again. So till then the chapter is actually over. So one more thing before saying goodbye, that now you can send your queries in our school app, that is CI Soft app. Okay, that you can send me your query in the chat box option, I think it is. So you can send me there, and whenever I'll see, I will respond it back. Okay. So now have a nice day. Bye bye. Take care.